Hi everyone, and again, welcome to Bio 101 online. The first lecture that we're going through for this course is an introduction to scientific study in biology. And so if you have a look at my PowerPoints, I'll be going through these with you as I dictate information about the PowerPoint slides. So the first thing I wanted to go over is the process of the scientific method. So in any area of science, scientists who are individuals who study something in nature, they go through a process called the scientific method. And the scientific method is laid out in this first slide that you can see on my screen. Here on the right-hand side, you see a picture of the scientific method. So the scientific method entails these several steps that are involved in this process. And this is to ensure quality of data, which is the evidence that you collect from asking some sort of question in regards to our environment and the world around us. So there are different types of scientists, those who study ecology, those who study evolution, those who study DNA and genetics, and they all follow this scientific method. So the first step in the scientific method, if you look at the first block, is to make an observation. So for example, when I was doing my graduate degree, I was studying in the field of DNA and genetics. And specifically, I was studying a disorder called fetal alcohol spectrum disorders. And so when I made an observation, I had to read scientific literature. And so scientific literature are specifically journal articles and books that have been vetted by experts that are in the field that have already published results within that area of study. So every field of study and every area of study has its own specific journal articles and information that you can find on it. You can find a lot of this information at the library's website, which I can go through with you after this slide. So I had to make an observation. I had to read several journal articles that had been published about my topic, which was fetal alcohol spectrum disorders. Now I focused on mice specifically because that was the model that we used in the lab, and so I had to read journal articles specific to mice and modeling fetal alcohol spectrum disorders in mice. So this takes a lot of work. For me, it took somewhere between 4 to 6 months before I was even ready to set up an experiment to test what I wanted to test. And so making an observation takes quite some time. You need to see what others have found in this particular topic in the field, and then you can actually design a question, which is the next step. So the next step is to ask a question. And mine involved, again, fetal alcohol spectrum disorders. So I asked which trimester results in the most severe effects in terms of the mice that are born that have been exposed to alcohol in utero. So I wanted to compare the first trimester, second trimester, and the third trimester. So I wanted to see how their brains were shaped and how that was different across 3 trimesters. So that was my particular question. And your question essentially forms the next step, which is a hypothesis. So a hypothesis is essentially something that answers that question. So in my example, I might have said something like the severity is the same across all 3 trimesters. So that would have been my hypothesis based on the literature that I read. So really a hypothesis is your best educated guess based on the scientific literature that you had observed in step 1 and based on your question. Now, the next step is to make a prediction based on that hypothesis, and these two can go hand in hand, making a hypothesis and then making a prediction based on that hypothesis. So you predict that you might see that all 3 trimesters, you see similar effects when you expose mice to alcohol in utero. Then you design an experiment. And designing an experiment, again, is based a lot on your first step, which is to make an observation and do your background research. So in my research, I had to do a series of behavioral analyses to be able to suggest that these mice exhibited features similar to those of patients who have fetal alcohol spectrum disorders. So I had to put the mice through rigorous testing that involved testing of developmental milestones, testing of anxiety, testing of hyperactivity, and then testing of learning and memory. Not only that, but I also looked at the brains of these mice. Specifically, I took out the DNA of the brains once they were adults, and I was able to look at the gene expression. So essentially, I was able to tell the mice who were treated with alcohol versus mice that were not treated with alcohol. I was able to compare them and say what genes are different in the mice that were treated with alcohol versus the mice that were not treated with alcohol. So I had a control and an experimental group. So that was my study design. So you always want to keep in mind what your study design is in an experiment. Usually there's going to be an experimental group where you apply some sort of treatment or test like the alcohol, and there's going to be a control group that you control against. So no alcohol, for example. And so you're going to test your hypothesis by running a series of little experiments that you'll put together into one big experiment. So like my behavioral tests, there were 4 or 5 of them, and then my DNA tests on the brains, and then all of that put together can essentially be analyzed, which is the next step. So you want to analyze your results. And so in an analysis, you have a lot of data points. So I had physical numbers that I got. So perhaps the mice that had undergone alcohol treatment in utero were taking longer in learning and memory tests. So I had actual numbers that showed me that the mice that were exposed to alcohol took longer to learn and to remember certain information. And the mice that were not exposed to alcohol, they had a standard amount of time for that. So nothing in biology means anything unless you actually analyze your results. So you could have lots of results, but unless you apply an analysis technique on them, it doesn't really mean anything. So an analysis technique involves statistical analysis, which is why biologists need to know a little bit about numbers and about stats. And so there are several different types of statistical analyses that can be done in order to analyze your results. We don't go into the different types of stats analyses, but just be aware that they must be done in order to make a conclusion. And essentially that's right here. So in the next step, you're making a conclusion about your hypothesis. So whether the hypothesis is correct, so did the data support your hypothesis? Or if the hypothesis is incorrect, so did the data not support your hypothesis? You don't necessarily say that your hypothesis was proven or not, because you can't really prove a hypothesis. So you say that a hypothesis is correct or incorrect, supported or not supported. And then once you have your information, you can publish your results in a scientific journal article. So that would be this body of scientific literature that others would look to if they were doing background research, like in step 1. So you want to add to the body of knowledge that's out there and available in your field of study. So you would write up your results and you would report them. And usually a report entails an abstract, which is a small summary of your experiment. And then it has several different components to it, like an introduction, which contains background research. It has your methods and materials that you used. It has your results, and it has your discussion or conclusion. Those are the 4 or 5 key elements to a scientific report. Now, it's okay if your hypothesis was incorrect. You can still publish, because even if you have those negative results, that still helps to add to the body of literature that's out there in your topic. So it's okay if a hypothesis is not correct, you can still publish. But sometimes if you are able to, you can go back and you can try again, reform your hypothesis, and then test it again. I wanted to also mention that reporting your results takes a lot of time. So this can take 6 months to a year to actually write up your results and then to send them to a scientific journal. So their scientific journals range from high impact, which are really good scientific journals, to lower impact, which are kind of easier to publish your results in because they're not as stringent. And so, especially if you're aiming high for a higher scientific journal, you can be put through a series of different steps that it takes to actually publish your work. And these steps can take a long period of time, like I said, 6 months to a year, and it can involve changing your experiment, repeating your results, altering information in your background, a variety of different things. So essentially, these are your 5 or 6 steps of the scientific method. You, again, start with background research or an observation, ask a question, make a hypothesis, form a prediction, do the experiment, analyze the results, make a conclusion, and then report your results. So those are the important steps to the scientific method. Now over here, I have a few points that I wanted to mention. So a hypothesis is different from a theory in biology. So you may have heard of theories in biology like the theory of evolution, the theory of the Big Bang, the theory of gravity. So how a hypothesis is different from a theory is that a hypothesis is a question that you're asking in a particular field that's kind of more narrow. A theory is quite broad. Theories have had several years, hundreds usually, hundreds of years of scientific reporting on them, and thus far there has been no evidence against those theories. That doesn't mean that it's not falsifiable. Everything in biology could potentially be falsifiable, but so far in the theories that are out there, there have been no reports against them. So theories are quite rigorous. They've had, again, years and years of backing to them, and there has not been reports against them. Hypotheses are more testable, more fluid, they're changeable, so they are quite different. And again, they're more narrow, they're a specific question versus a theory that's much more broad, like again, the theory of evolution. A hypothesis would ask a specific question about natural selection for, again, on a specific species. So again, that's where you're getting more into a narrow question. Now, the scientific method must have evidence, and that's where your experiment comes in, and you get your results and you analyze them. That is what evidence is. Again, the data that you collect needs to be verified through statistical analyses. Something you want to control for in your experiment when you're designing it are conditions and variables. So for example, in my study, which was on fetal alcohol spectrum disorders and using mice to model them, I had to consider, by doing my background research and making observations, I had to consider what variables I needed to control for in my experiment. For example, when I looked at the pups are baby mice, that's what they're called. When I looked at the pups and was assessing their behavior, like anxiety and activity levels, things I had to control for were, am I using males or am I using females? What's the weight of the mice? Even the pregnant mothers initially, where I was giving ethanol versus no ethanol treatment, I had to make sure that everything was standard. Same food source, same lighting in the room, same temperature of the rooms. So anything that could potentially explain the results must be controlled for, and these are called variables. Again, you can never prove a hypothesis is true. We don't say that in scientific literature. We don't prove hypotheses because you can never prove something absolutely. You would have to test it in every scenario on the planet in order to be able to prove it. So that's very difficult to do. It can't essentially be done. So that's why we say a hypothesis is either supported or not supported by the data. Again, that's what brings the difference of something that's scientific versus non-scientific. So scientific is something that follows the scientific method versus non-scientific. And so, for example, the internet is an excellent source of literature for us, but you want to be careful about finding things that are specific or scientific versus non-scientific. Remember, everything in biology is falsifiable. We could always find data, years from now or weeks from now that something was actually not supported where before we thought it was supported. So anything can be falsifiable because of the nature of the scientific method. Something to keep in mind along with those lines is reproducibility. So when I was making my background research and observations, I had to make sure that I was able to use experiments that had previously been used before for studying the same things I wanted to study. So it doesn't help to add to the body of literature if you are designing experiments that have never been done before using equipment that's never been used before. You want to follow and make sure you're using reliable experiments, and that points to the next point as well. Back to the reproducibility of your results, it also is important that if someone were to repeat your exact methods, that they're able to get the same results. So for example, several years back, there was a study done on red wine, and red wine was thought to be good for you to consume because of a component called resveratrol. And so there was a scientist who took mice and he gave them resveratrol, and he found a 30% increase in lifespan extension. So that means living 30% longer in mice who had these high doses of resveratrol, which again can be found in red wine. He brought this to a pharmaceutical agency, and so he packaged these pills that were known as resveratrol, or perhaps by another name. And so he packaged these resveratrol pills, and he was able to make millions and millions of dollars selling them as a lifespan extension technique. However, when other scientists, other groups around the world who wanted to repeat his results, because as scientists, that's what we like to do as well. We want to make sure all experiments are repeatable and hence valid in the literature. If you repeat it, you should get the same results. So other scientists around the world repeated his results and did not find the lifespan extension that he had suggested was in fact there. And so later on, he was hearing about these other scientists repeating his research. So he himself went to repeat his research. And when he did not find the lifespan extension by 30%. He found it was the same across a control versus resveratrol mouse. And so he published that instead, but it didn't really matter because he had made millions of dollars anyway. So that's something to keep in mind. You want to make sure that when you're reading scientific reports, that the methods are valid, they've been used in other experiments, and that the results are actually reproducible, so that other groups around the world who have tried this method, that they found similar results. So that's what I wanted to say about the scientific method. I also wanted to take you through and show you our library website. So if you go to sc4.edu and you scroll down and you click on library, this is an area where you can be sure that you're finding good sources of information that are valid and reproducible and that are not biased. So oftentimes when you just Google search, for example, you can find biased sources of information. And so anytime you're searching for information, you can use this search tool here and look up information about your topic. So for example, if I was looking up climate change and threats to biodiversity, I would write climate change, and it's in quotations because it's more than one word. And then I would capitalize and write and I would put biodiversity. And biodiversity is one word, so it doesn't need the quotes. So then I click find it, and that brings you through the library's webpage into lots and lots of information on climate change and biodiversity. And you can actually click over here on the left-hand side, scholarly and peer review, And then journal articles if you're interested in those journal articles, which is where new information gets published usually. You can also look at books if you're interested as well. You can look at the time frame. So let's say I just want the last 12 months and I want to know what's out there in the literature. And there's other things that you can customize as well. But for example, there's several articles that come up on climate change and how that affects biodiversity. And that's important because we will delve into ecology fairly soon. So climate change is an important topic we'll get to. But if you're looking for scientific reports, this is how you would find them. Versus if you went to Google and if you typed in the same information, climate change and threat, or climate change and biodiversity, you would find biased sources of information oftentimes. So news articles is usually what comes up, lots of different news articles and different web pages that can potentially be biased, which means there's certain groups or certain individuals writing these articles or blogs that give you information about the topic. But something you want to keep in mind is you want to look at their sources, so their references that you use to make the claims that they have on those websites. So that's something to keep in mind as well. Going back to our discussion, something you can do is actually go through the library's website and you can click on one of those scientific articles that I mentioned, like climate change and biodiversity, and see if you can skim through it and find some of this information in it. So specifically, questions that you would want to ask yourself based on our short discussion was, what was the purpose of the study? Why did the researchers want to investigate climate change and biodiversity? What was the hypothesis? So sometimes researchers will say, we hypothesize that, but sometimes they won't, and it's left for you to interpret what their hypothesis was. Identifying variables that you had to control for. So this is where the conditions and variables are important. If you're looking at climate change, if you're looking at studying biodiversity, what do you have to control for? And can you control for those? So being able to control for variables and actually being able to actually control for those variables and being able to say you control for the variables are two different things. Was there a control group? Was there a treatment group? And in something like ecological studies that address climate change, you probably won't see control and treatment groups. This is more so for different types of trials. Usually, if you think about drug trials, there's a control group and a treatment group. That's more so for what that's entailed for. See if you can find one or two key results from the study. So that would be in the results section. Were the results confirmed using statistical analysis? And you don't have to know what type of test, but just see if you can figure out if the results were confirmed using a stats test. And lastly, do the data support the hypothesis? So you can find that usually in the discussion or conclusion. And sometimes you may need to infer. It might not be spelled out for you based on the scientific article, but sometimes you will need to infer what the data says. So something I wanted to move on to in this discussion of an introduction to biology are properties and characteristics of life. So if you think about life and all things that are living, which we call biotic, we call non-living things abiotic. And so when you think about living things, what do they all have to have in common? So response to stimuli. They have to be able to respond to some sort of stimulus. Evolution. So that is a property of life. And evolution means being able to adapt to your environment. That's essentially the meaning behind the word evolution. If you can change either your physiology or your DNA, if you can change by adapting to your environment, and those changes are passed on in your offspring or your children, then you are undergoing evolution. A great example of this is the evolution of bacteria for antibiotic resistance. Order or cellular organization. So you have to be made up of cells. That's a requirement of life. So if you think about different living organisms, bacteria are made of cells, plants are made of cells, humans and other animals are made of cells. Interestingly, viruses are not considered living things because they are not made up of cells. They're just made up of two things, a protein component and a DNA or RNA, which is a similar material to DNA, in that component. And so those two components together make up a virus. So they're not considered living things just because they're not made up of cells. And we will discuss what cells are and the intricacies of cells a little bit later. Regulation. You have to be able to regulate yourself, and along with that, maintain body homeostasis. Um, Along with that, you have to be able to process energy. So plants get their energy through a process known as photosynthesis, and humans and other animals get their energy through a process known as cellular respiration. Reproduction, so whether it's asexual by yourself or sexual two individuals, you have to be able to reproduce. Some bacteria can undergo both processes depending on their environment, and you have to be able to grow and develop. So these are largely the properties of what makes something a living thing. Now, if you ask me the question, or if I ask you the question, has anyone ever invented synthetic life? And the answer is yes. So there's a man by the name, a researcher by the name of Craig Venter, and he created what's called the minimalist genome back in 2010, which is almost 10 years ago now, but he was the first individual to create a synthetic life form. So essentially what he did is he took an organism that already existed, like a bacterial species. He removed the genetic information in that bacteria, and then he built the chromosomes, which is how our DNA is wrapped, right into that shell of that organism. And then he transplanted it into another organism. And so this was his version of a synthetic life form. And if you think about how many genes a human has around 25,000 genes. Bacteria have 1,000 to 3,000 at their smallest forms. And so he created a synthetic life form like bacteria with the smallest genome, smallest number of genes, which is why it's called the minimalist genome. And it was around 400. So he built this organism all on its own, and within the DNA, he actually put his name into it. So if anyone were to ever sequence that DNA, which is figuring out the letters of the DNA, if you've learned those before, they are A, C, G, and T, and we'll talk more about them later. But if you were ever to sequence it, you would actually find his name there. So all of life is organized into a hierarchy. So we have something at the bottom and something at the top. And so this is the hierarchy of life from your book. So organelles are at the very bottom of the hierarchy. So they are right here. So organelles make up things called cells. And cells are the basic units of life. So organelles are the little components inside of a cell that makes up the cell. So like a nucleus, if you've heard of that, for example, that contains DNA. That's an organelle. And so organelles, several of them, make up cells. Several cells put together into a working unit make up a tissue. Tissues put together make up organs and organ systems, which are organs that function together in the body. And then these organs and organ systems, when you put them together, they make up an organism. Now, when you take two or more members of the same species and you put them together into a group, that makes up a population. And populations, when you take two different species and put them together and have them interact with each other, that's called a community. And then when you build on top of that community by adding abiotic factors, so non-living things like sunlight, soil, nutrients, when you put all of that together and several different species together, that makes up an ecosystem. And then all of the ecosystems on Earth together make up the biosphere. Biosphere is at the top level of the hierarchy of life. Now, these words here, organism, population, community, and ecosystem, when we talk about ecology, which is after we discuss evolution, you'll see how these 4 levels make up the levels of ecology. So these 4 words become important when we discuss ecology. And all of life is organized into something called the phylogenetic tree of life. And the phylogenetic tree of life, nothing of which here you necessarily need to memorize. You will not be asked to redraw this tree, but you should know that all of life is categorized in 3 categories: bacteria, archaea, and eukarya. So these are what's called the 3 domains of life. And animals, we fall into animals, so we're humans. We fall in right here on this tree of life. An ancestral common ancestor is way down here. And then over evolutionary time, so going from the bottom to the top is evolutionary time. Over time, different lineages split into different branches, and these 3 branches are called the 3 domains of life. Again, animals fit in here on this lower branch to the right. Again, this red group is called archaea, and then this blue group is called bacteria. So bacteria have their own domain of life. Archaea have their own domain of life. Archaea, in terms of their cellular organization, are really similar to eukarya, but they are actually function more as a bacterial species. So they ended up having their own domain of life. And then again, eukarya fall into this category on the right that's brown. And within eukarya, we have things like animals, fungi, plants, protists, other organisms that are usually multicellular. And so that's it for our introduction to biology. If you have any questions about this lecture, please feel free to email me. Thank you.